Imagine your immune system is this vast, just incredibly efficient manufacturing factory.
Right.
Like year after year, from the day you're born, it flawlessly produces billions of specialized cells. You've got your elite special ops forces to fight complex viruses, your standard infantry for everyday bacterial threats, and, your medics to patch up physical wounds.
Perfect balance.
Exactly. But then as the years go by, something inside the factory subtly shifts. What happens when the upper management suddenly decides to stop producing those elite special ops forces?
Oh, it's a disaster.
Right. And instead, they start obsessively churning out first responders and medics constantly, round the clock, even when there's absolutely no emergency happening.
Well, the whole system becomes incredibly unbalanced.
Oh,
The factory is still running at full capacity, burning through energy and resources, but it's producing completely the wrong inventory for the actual reality on the ground.
yeah.
You end up with a massive misallocation of resources that leaves the whole organism totally vulnerable.
And that bizarre misallocation is the core focus of our deep dive today. We are unpacking a truly groundbreaking study published in Nature Cell Biology.
It really is a phenomenal paper.
It is. This paper manages to reveal the exact, incredibly granular molecular mechanism behind why our immune systems age. We're going to explore how a very specific elite subset of blood stem cells gets epigenetically reprogrammed by the everyday wear and tear of inflammation. And more importantly, how that reprogramming leads to a whole cascade of vulnerabilities as we get older.
Which is something that directly impacts every single person listening to this right now. Because this isn't just, abstract microscopic biology happening in a vacuum.
No, not at all.
This internal factory shift is the exact reason why, as we age, we become so much more susceptible to everyday pathogens. It's the underlying mechanism explaining why older individuals often have much poorer responses to vaccines.
Yeah, that's a huge issue.
It is. And why they face elevated risks for chronic inflammation, cardiovascular disease, neurodegeneration, and even cancer. We are literally looking at the root cause of why our biological defenses break down over time.
So to fully understand how the factory breaks down, we have to start at the very top of the supply chain, right? We need to talk about hematopoietic stem cells,
Right.
or HSCs.
Right. The HSCs.
Those are the absolute ultimate source of all blood and immune cells throughout our entire lifelong journey.
Yeah, they are the absolute apex of the hierarchy. Every red blood cell, white blood cell, and platelet in your body can trace its lineage back to these stem cells residing in your bone marrow.
Wow.
Now, in a young, healthy system, these stem cells maintain a beautiful, highly regulated balance. They produce a steady, appropriate ratio of what we call myeloid cells.
Wait, let me just pause you there so we can define that. Myeloid cells, those are basically the infantry we talked about.
Exactly. Myeloid cells are your innate immune cells, things like macrophages and neutrophils, basically your biological first responders. They deal with immediate generalized threats.
Okay.
Got it.
And alongside those, they also produce megakaryocytes, which are the cells responsible for creating platelets for blood clotting.
So the medics of the factory.
Right, the medics. And crucially, alongside the infantry and the medics, they also produce a steady stream of lymphocytes.
And those are the T cells and B cells, right?
Yes, your T and B cells. Those make up your adaptive immune system. They are the targeted special ops forces that remember specific viruses from years ago and hunt them down with incredible precision.
But the research paper details this massive problem that happens as we age, a phenomenon they call lineage skewing. Basically, as we get older, the factory becomes functionally impaired.
Severely impaired.
Right. It starts overproducing those myeloid first responder cells and the megakaryocyte medics at the direct expense of the lymphocytes. Let me make sure I'm visualizing this right.
Go for it. Is it kind of like if a city suddenly panicked and reallocated its entire civic budget to hiring thousands of paramedics and traffic cops, but to pay for it, they completely fired all the specialized detectives who actually solved the complex existential crimes?
That is a perfect analogy. And taking that city analogy a step further, the reason the mayor of the city makes that terrible budgeting decision is deeply tied to a concept known in longevity science as inflammaging.
Inflammaging's just chronic inflammation from aging.
Exactly. It's the chronic low-grade inflammation that typically develops in all of us as we get older. The sources show us that this lineage skewing, this budget reallocation, isn't just random wear and tear or passive damage to the stem cells.
It's intentional.
Yes, it's an active structural shift in the cell's output. The body is constantly sensing this background noise of inflammation, so the factory management basically panics.
I see.
It thinks it needs to constantly patch wounds and fight immediate, generalized bacterial threats. So it ramps up production of platelets in innate immune cells, completely neglecting the long-term, specialized defense required to fight off novel viruses.
Which leaves you incredibly vulnerable because you have no detectives left to identify a new threat when it arrives. It really makes you think about your own body, right?
Absolutely.
Like, this is the exact microscopic reason why a common cold that took you 3 days to beat in your 20s suddenly lingers for 3 or 4 weeks in your 60s. The stem cells at the top are skewing their output because of this background noise, which fundamentally changes how an older body responds to everyday stress.
Right. It's a complete shift in strategy, but an unhelpful one.
Okay, so let's do a quick recap for you listening, just to solidify this first major concept before we dive deeper. We're going to do this throughout the deep dive.
Good idea.
So, bullet number 1. HSCs, or hematopoietic stem cells, produce all our blood and immune cells. Bullet number 2. Aging causes this lineage skewing toward myeloid and megakaryocyte cells and away from lymphocytes. And bullet 3, this directly causes age-related immune decline.
Perfect summary.
So here is a question for you listening to ponder. Have you ever noticed how your body's healing process and your susceptibility to lingering colds seem to fundamentally change as people get older? This factory imbalance is exactly why.
Yeah, that realization is what drove the researchers to the next critical phase of the study. If the whole system is skewing toward these first responder cells, it makes you wonder who is actually giving that skewed order.
Right. Who is the manager making the bad calls?
Exactly. They needed to find the specific management tier responsible for the breakdown. And they didn't just guess. They used incredibly advanced techniques to map the hierarchy.
The paper mentions they use something called single-cell RNA sequencing on mouse stem cells.
Yes, is cRNA-seq.
For anyone who hasn't spent time in a genetics lab, let me break that down. Instead of taking a whole smoothie of tissue and guessing what's inside, single-cell sequencing is like pulling out every individual piece of fruit from the blender and reading its specific cellular instruction manual.
That's a great way to put it.
It tells you exactly which genes are turned on or off in one single cell at any given moment.
It provides an unprecedented level of resolution. By reading those individual instruction manuals, the researchers were able to map out the developmental trajectories.
Meaning they could see how they grow up.
Right. They could track exactly how these stem cells mature, differentiate, and make decisions. Through that mapping, they found the absolute developmental apex, the true boss of the factory.
The big boss.
The big boss. It's a specific, highly elite subset of stem cells they identified as Meg3 positive HSCs.
And the source material gives this incredibly long molecular barcode for these cells. They have specific surface markers that allow scientists to physically isolate them. Things like CD150 high, SCA1 high, and a whole alphabet soup of other tags.
Yeah, CD24 high, CD201 positive, it's a lot.
But the key takeaway for us is that these markers act like an ID badge, right? Scientists can use that badge to pull these exact boss cells out of the bone marrow and study them in total isolation.
And what happens to these specific Meg3 positive boss cells as we age is genuinely fascinating. In youth, they are the gold standard. They have the highest reconstitution capacity.
Meaning what exactly?
Meaning if you were to take just a few of these young cells and put them in a completely empty bone marrow factory, they could rebuild the entire immune and blood system perfectly from scratch.
Oh, wow. Just a few of them.
Just a few. But starting in middle age, something strange happens. The population of these specific Meg3 positive cells expands dramatically. They multiply.
But wait, let me pause you there. Here's where it gets really interesting. If these Meg3 positive cells are the ultimate bosses, the absolute top of the food chain, why does having more of them in middle age break the system? Shouldn't a larger management team make the factory more robust and capable?
You would think so, yeah. But it actually causes a breakdown because the quality of the bosses degrades. Their internal instructions fundamentally change. To prove this, the researchers performed transplantation experiments. They took Meg3 positive stem cells from middle-aged mice, which is about 8 months old in mouse years, and transplanted them into recipient mice to see what they would build in a fresh environment.
And what happened?
The middle-aged cells had a drastically altered potential. They generated a massive surplus of those CD11b positive myeloid first responder cells, but severely neglected the CD3 positive and CD19 positive lymphoid cells.
There was also a highly specific detail in the paper about the T cells that actually did get produced, The few special ops forces that made it off the assembly line were essentially coming out broken, right?
Yes, they were getting arrested at a premature developmental stage in the thymus. A healthy T cell needs to develop specific receptors, known as CD4 and CD8, to function properly and hunt down viruses.
But these didn't.
No, the middle-aged boss cells couldn't push them past that infantile stage. We call it a CD4 negative, CD8 negative stage. The stem cells lost their self-renewal capacity and became inherently biased toward making megakaryocytes and myeloid cells.
And speaking of megakaryocytes, the cells that act as the medics and make platelets, the researchers uncovered this incredibly cool detail about their behavior, specifically regarding something called ploidy.
Yes, ploidy is fascinating.
Normally, a human cell has 2 sets of chromosomes, right? But megakaryocytes are totally unique. They intentionally gorge themselves, copying their DNA over and over again without actually dividing into new cells. They just get larger and larger, bloated with genetic material.
Yeah, exactly. And the study found that young Meg3 positive boss cells primarily make intermediate-sized megakaryocytes. We're talking 4N to 8N, ploidy, 4 to 8 sets of chromosomes.
Okay.
And the main job of those intermediate cells isn't actually to dump out platelets into the bloodstream. It's to stay in the bone marrow and physically support the structural niche of the factory itself.
Oh, so they are the maintenance crew keeping the stem cell environment healthy.
Precisely. But when we look at the middle-aged boss cells, their output is completely warped. They start churning out these massive, bloated megakaryocytes, 16N to 32N ploidy, whose sole biological purpose is to dump massive amounts of platelets into the blood.
And they also make 2N1s, right, for immune modulation?
Yes, the 2N1s as well. So they completely abandon the structural maintenance of the bone marrow factory just to churn out emergency medics and immune modulators. It's a molecular demonstration of the system breaking down from the very top.
The bosses expand in number, but they lose their balanced touch.
Exactly. They stop supporting the factory infrastructure, they fail to develop specialized T cells, and they obsessively overproduce platelets and first responders.
So let's do our second mini recap for the listener. Bullet 1. Meg3 positive HSCs are the apex stem cells that orchestrate blood production. Bullet 2. They expand wildly in middle age but lose their balanced touch. And bullet 3. They start overproducing specific types of platelets and myeloid cells while failing to support T and B cell development.
Spot on.
So here's a question for you. If you could reprogram one boss cell in your own body to act young again, how much downstream damage do you think you could prevent?
Well, that exact train of thought is what led the researchers to the next crucial phase of their investigation. We know the Meg3 positive cells go rogue. We see the output changing. But why does it change?
Right.
If the underlying DNA sequence, the actual genetic code, is the exact same in a middle-aged cell as it was when that cell was young, how do the instructions change so drastically?
And this is where we enter the world of epigenetics.
Yes.
The researchers threw every advanced molecular tool they had at these cells. They used multi-omics profiling, things like ATAC-seq, CAT-and-tag, LC-MS-MS. They looked at how tightly the DNA was spooled up to see which genes were physically accessible to be read.
They didn't just look at the code. They looked at how the cell was physically interacting with the code.
Exactly. And through that massive sweep of data, a glaring culprit emerged. They found a huge accumulation of a specific histone modification, an epigenetic mark called H3K23SC.
Yes, in the middle-aged Meg3 positive cells.
To help visualize this, we shouldn't think of the DNA as a static book. Imagine the DNA blueprint for the factory is spread out on a massive table. This H3K23 ACK mark is essentially a bright neon sticky note placed on very specific pages of that blueprint.
I love that analogy.
It's a chemical flag that screams to the cell, read me and build this immediately.
It really is. But the researchers needed to know what was placing the sticky note there in the first place. They traced it back to a specific trigger, environmental inflammatory signals.
Like what kind of signals?
Things like IL-1 alpha, or bacterial mimics like LPS, or viral mimics like poly-IC. When the body experiences this low-level inflammaging, it activates a specific pathway in the stem cell. It triggers a pioneer transcription factor axis known as P65RELA-CAT6A.
Okay, that's a mouthful. Let's give the listener a mnemonic to remember this whole chain reaction.
Good idea.
Let's use IHTP. IHTP. So the I stands for inflammation. The background noise of aging hits the cell. That triggers the KAT6A enzyme, which then walks over to the blueprint and slaps that neon H onto the DNA, the histone mark, H3K23AC.
Right. And crucially, when the researchers looked at where those neon sticky notes were being placed, it wasn't random. The data showed these marks were being slapped directly onto specific genes, genes like GP5, HOXA7, TRIB1, and ES2.
And what did those genes do?
They are heavily involved in forcing the cell down the megakaryocyte and myeloid differentiation pathways.
So genes that specifically command the overproduction of medics and first responders.
Yes. The everyday inflammation is literally rewriting the daily work orders of the stem cell. It's bypassing the normal checks and balances and hardwiring a bias into the cell's output.
Okay, recap time. Bullet 1. The root cause of the shift is epigenetic specifically, an increase in the H3K23 YSIC mark.
The sticky note.
All right, bullet 2. This mark is triggered by environmental inflammatory signals via the P65KK-TSA axis. And bullet 3. H3K23AC acts like a neon sign directing the cell to make more myeloid and MK cells. So question for you listening. Knowing that everyday inflammatory signals can literally rewrite your stem cell's epigenetic instructions, how does that change the way you view stress and chronic inflammation? It's not just a feeling, it's a chemical signal tagging your blueprints.
It's a very sobering thought. But to fully understand how this misdirection happens, we have to look at how that sticky note is actually executed on the factory floor. Because the histone mark, a chemical sticky note, doesn't actually do anything on its own.
Right, it's just a piece of paper.
Exactly. It's inert. It needs a reader protein to come along, see it, and take action.
Which brings us to the next letter in our mnemonic, T, the executioners of the process. The source material introduces a reader protein called TIM24.
Yes, TIIM24.
You can think of TIM24 as the middle manager walking the factory floor. The data shows that TIM24 is physically drawn to and recruited by those H3K23-ACK neon sticky notes.
And once TIM24 binds to that mark, it executes the final step of the cascade. It significantly enhances the transcriptional activity of PU.1.
And there is our final letter, PU.1, so IHDP. Inflammation leads to the histone mark, which recruits TIM24, which activates PU.1.
Exactly. And PU.1 is basically the foreman on the factory floor shouting the actual orders at the assembly line. It's a highly powerful transcription factor that acts as the final executioner in this whole process.
So once PU.1 gets involved, it forces the cell to fully commit to becoming a megakaryocyte or a myeloid cell.
It locks in the fate of the cell. Once PU.1 is hyperactivated at these specific gene sites by the middle manager, the cell crosses a point of no return. It can no longer become a specialized lymphocyte. It is permanently committed to the first responder or platelet pathway.
But I want to make sure we aren't mischaracterizing this. So if H3K23XE is the sticky note, TRIM24 is the middle manager who reads it, and PU.1 is the foreman shouting the orders. Is PU.1 the bad guy here? Or is it just a normal worker following corrupted instructions passed down from the inflammation?
I'm really glad you asked. That is a vital distinction. PU.1 is absolutely essential for life and for normal, healthy blood development. It is not a villain.
Okay.
But in these aged rogue stem cells, the specific physical interaction between the neon sticky note and the middle manager causes PU.1 to become hyperactivated in a highly localized, biased way.
So it's the context.
Right, it's the context that makes it destructive. The inflammation corrupts the management chain, which forces the foreman to execute skewed orders, effectively locking in that aging immune profile we discussed at the start.
Let's quickly recap that chain of command. Bullet 1. The H3K23-ACK mark recruits the reader protein TRM24.
Yes.
Bullet 2. TRM24 boosts the activity of the transcription factor PU.1. And bullet 3. PU.1 executes the skewed factory orders, locking in the aging immune profile.
Perfect.
So here's a question for you listening. Can you think of a middle manager in a system you know that completely alters the outcome of a project just by how they read the initial instructions? Because that's exactly what's happening inside your bones right now.
It's a great analogy, and it brings us to the most exciting part of this entire deep dive.
Yes, the rescue mission.
Exactly. If we now understand the exact molecular chain of command, from the inflammatory trigger to the sticky note to the middle manager to the foreman, can we intervene? Can scientists actually reverse the clock on this process?
And they did, right? The researchers experimented with disrupting this exact chain in the lab.
They did. They knew they needed to disrupt that chain. So they used a specific chemical inhibitor called IACS 9571. And in another experiment, they used a TRIM24 bromodomain mutant.
Basically a genetic tweak.
Right. And what both of these interventions did was highly specific. They acted like a molecular pair of scissors to block the physical interaction between the neon sticky note and the TRIM24 middle manager.
So they essentially cut the wire. They took away the middle manager's ability to read the bad instructions.
Exactly. And the results detailed in the paper are stunning. By preventing the manager from reading the sticky note, they successfully suppressed the hyperactive formin, Pu.1.
Wow. And that actually fixed the factory output?
It did. When they applied this intervention to the aged stem cells, it successfully restored a balanced lineage output. The factory stopped its obsessive churning out of myeloid cells and megakaryocytes and actually went back to producing the healthy, targeted lymphocytes.
They literally unskewed the factory. That is incredible. We can't necessarily stop the environmental inflammation from occurring in an older body. The background noise is still there, but by blocking that one specific interaction, the cells revert back to a youthful state of production.
Adding to that, the intervention also significantly reduced the inflammatory signals coming from the cells themselves.
What do you mean?
Well, older cells often develop what's known as a senescence-associated secretory phenotype, or SASP. They basically sit there and leak inflammatory signals, making the whole environment worse. This intervention quieted that down.
That feels massive. It proves that immune decline isn't just a permanent, irreversible wearing out at the parts.
It highlights incredible therapeutic potential. It proves that age-related immune decline is largely driven by an active epigenetic program. It's a temporary set of instructions that can be uncoupled.
Reversing the clock.
Exactly. It strongly suggests that if we can develop a way to target this specific interaction safely in humans, we could potentially rejuvenate aged immunity.
Let's do our final mini recap. Bullet 1. Disrupting the physical connection between H3K23-axis and TIM24 stops the skewed signaling.
Yes.
Bullet 2. This intervention restores youthful, balanced blood cell production. And bullet 3. It proves that age-related immune decline is a reversible epigenetic program.
Couldn't have said it better.
So a question for you. If scientists can eventually package this TIM24 blocker into a safe therapeutic pill, would you take a pill that resets your immune system's age? It's wild to think about.
It really is. To synthesize the journey we've taken through this research, we started at the absolute top of the cellular hierarchy with a single elite stem cell. We watched how middle age and chronic inflammation caused those cells to multiply but fail in their critical balancing duties.
Right.
We zoomed all the way down to a single molecular sticky note placed on the DNA. We saw how a middle manager read that note, hyperactivating the formin, which then doomed the cell to overproduce first responders at the cost of our adaptive immune system.
And finally, we saw that blocking that one specific physical interaction could turn back the clock and restore a youthful balance. It's just an incredible piece of science. And I want to leave you with a final paradigm-shifting idea to mull over as we wrap up this deep dive.
Let's hear it.
We have been conditioned our entire lives to think of aging as inevitable entropy. We picture it like a rusted car falling apart over time. We assume the hardware is just degrading and there's nothing we can do about it. But what if a massive portion of our physical decline isn't rust at all?
That's the big question.
Right. What if it is a perfectly functioning, highly efficient factory that is simply following a stubbornly pinned, completely wrong sticky note? If aging is driven by an epigenetic software glitch rather than a permanent hardware failure, then the fountain of youth might not require rebuilding the machine from scratch. It might just be a molecular software patch.